All right, well, thanks everybody. That was a really fantastic session. I know I've got a ton of questions, so I'll kind of get us started. But if anybody has questions for any of the speakers, come on up to the mic. One thing I'd really like to know is, and maybe we'll start with Julia, because you kind of began to talk a lot about interacting with clinicians and thinking about public health policy. What are the most important facts about the microbiome or findings about the microbiome that we really need to make sure that clinicians and policymakers know?
So, in that regard, I think it's sort of you want to generate the excitement, but you can't really promise. But I think the microbiome has the potential to be a biomarker in the same way when you have diabetes and you prick your finger and test your blood sugar levels. In the same way as that, we could use the microbiome to anticipate. The clinical trial that I run is for kids who have bad eczema. When is the kid going to have a flare? And start the treatment before you've gotten to the excoriating rash. I mean, so I think that we do have lots of treatments for these diseases, and it's about trying to get early intervention. And also for some of these, we could get to the point where we say, well, here are the 5 things you could try for this. Well, which one is really going to work? And if we aren't finding the signature completely in the human genome, then maybe part of that is how you respond to certain drugs has to do with what is your existing microbiome and what's your metabolic potential. So I think it's really about using the microbiome as part of personalized medicine. I mean, in terms of policy, there's so much. I mean, I think that it's great if people start to think about themselves as superorganisms and lose the language of warfare, try to start thinking about, living in balance with the microbes. And from a policy perspective, I mean, we really need to get out ahead on regulating antibiotics and antibiotic stewardship and, make sure not to lose that, make sure to really, understand what are the risks of living so closely with all these microbes.
Yeah, thanks. Peter, any thoughts on policy or clinical applications? What do folks need to be thinking about?
Yeah, I mean, I guess I agree with a lot of what Julia said. I would maybe, broaden it a little bit in that, we think that the microbiome is relevant not only for medicine, but also for sort of daily aspects of health, such as our diet and nutrition. And so understanding sort of which particular substrates are most likely modified by the microbiota and how variations person to person will predict whether or not you are able to metabolize a given diet or produce certain bioactive compounds that might have important implications for diseases like IBD or cancer is a really important way forward. And, I think there's been a lot of interest in the past. Eric talked about Activia, and, there's obviously many products out there. But, understanding the science to a level where we can actually sort of support these products, not with the goal of supporting them, but understand the mechanistic basis of some of these interventions and make more concrete recommendations to people in terms of diet and health, I think, is a big one of the main focuses moving forward.
Larry, you don't have any strong opinions on this point, so maybe we'll go right to David. But maybe you want to chime in.
The only thing I'd say is you all take it for granted that people know that there's a gut microbiome, for instance. Everywhere I go and talk about this, people said, how come nobody told me that there were 10 times as many cells? We're deep, deep behind in education and at all levels. And I think, actually, the more I think about this thing, the more we ought to be humble and say, well, maybe we should just work on the microbiomes and occasionally sprinkle a little human in if we're only 1% of the genes.
Yeah, I would agree with all that's been said. There are all kinds of potential opportunities for intervention, and Peter's alluded to some of those. I don't think we begin to know how and where we might most effectively intervene or monitor. There's the whole issue of small molecule discovery. We have tended to look almost everywhere but within for what might be the next previously unrecognized, incredibly important small molecule or drug-like compound that is probably already been evolved and adapted for human activity within the system. And I guess in the end, some of you know Brendan Bohannon, but he's made analogies of the following kind: that if you were a park ranger and your responsibility was a national park, what's the first thing you want to have? It's the park management plan. And that's basically what we need for this park.
Great. Nitin?
Yeah, so is this on? So we've been looking primarily at populations of microbial distributions, especially for the gut microbiome. And so I have a two-part question. One is, to what extent do you think variation plays a role? So you may have the same population distribution, but different variants of the population that may be colonizing your gut. The second part is the spatiotemporal structure of the gut microbiome is really difficult to get at. So do you think there are imaging technologies that you think that might be of use once you figure out the population variations, but that has to be customized to each individual?
I'll just start by alluding to something that's already been described, which is despite the fact that we like to lump things together, there is some really critical variation at the level of strains, right? And they've been described well in sort of in the context of lateral gene transfer, but blocks of genes even intersperse little critical elements, and this is where the work that I was hoping one of us might be able to describe that was done with Jill just highlights some of the variation that takes place, say, between two Citrobacter strains of the same species in the developing creamy gut, where the difference is several dozen genes and sequence differences in intergenic regions that may correspond to regulatory RNAs that are regulating in turn suites of other genes and their activities. And these are organisms that are going to be identical on a plate that never would have been resolved, and whether they mean something and their shifts in abundance over short periods of time, we won't know until we begin to do this kind of thing on a more regular basis.
I noticed that Julia just said that we've got to be awfully careful about the use of antibiotics and the overuse of antibiotics. And yet, when I saw your presentation, I was looking everywhere for some quantification of the immune status, particularly the innate immune status of the patients. Because there are two factors in whether an antibiotic will do its job in vivo, as distinct from in vitro, where pretty well all the work is done. And one of those factors is the innate immune system.
Right.
And now that we have that data that was published last week from the cave in Africa, showing antibiotic-resistant species a million years old, never exposed to antibiotics, I can't help thinking that we've got to be very careful in not overreacting and saying, oh, too much antibiotics, too much too many resistant species, because what is happening in general is that innate immunity in Homo sapiens is getting worse decade by decade.
So thank you. Actually, so the slide after my acknowledgments is the clinical state of all the patients, and you're right. The majority of patients who were attacked are on immune suppressants because they'd just gotten a transplant. And so that the innate immune system, I mean, for many of these bacteria, the immune, the multi-drug resistant bacterial bacteria, the innate immune system could do their job if we would give them the chance. But an increasing, not only maybe if the innate immunity is, I don't know how how you're stating that the innate immunity is decreasing, but we're putting an increasing number of patients on immune suppressants as we do, matched related donor, matched unrelated donor, transplants. So, and and also we see this problem in long-term care facilities because elderly patients are, are increasingly immune compromised. So, and then we have an increasing number of, patients who are HIV positive. I mean, we as a population are medically managing many risks that are putting people as immune compromised when that isn't probably part of how our genetic programming was created. That's a medical risk that we're putting people under for a good reason, but now we have to manage that risk.
Right. Is this still on? The data on human innate immunity, if you look at the exponentiating rates of the autoimmune diseases, it tells you right there that what's happening decade by decade. Well, it may not be accepted in these circles, but in the autoimmunity conferences I go to, it's now recognized that innate immunity is at the key of the autoimmune reaction.
So can I hear your opinion on the hospital of the future where, for example, patients check in and they get a device to track them through the hospital, and medical practitioners are constantly being tracked as to where they are and who they're interacting with, real-time sequencing of sampling to basically be able to real-time and analyze these outbreaks? Is that a distant future thing? Is that even practical?
So I have a physician friend who's a cardiothoracic surgeon, and he says that he used to be a jet engine pilot fighter, and now they got all these checklists they want him to go through, and he says he feels like a bus driver. And then he'll look at me and he'll say, but you know what, my patients have a better outcome, so what am I going to do?
But that's like something they have to do, but it's tracking them around. I mean, you know exactly where they are. There's a system doing a systems analysis of the hospital in real time to predict when outbreaks will occur, and if they do, to backtrace it to find where the problem is.
Right, and I do think that the best hospitals are really good learning organizations, and you will start to see things like, you will start to see things like that where hospitals start to really track, and they already do. What are their weaknesses? What are their risks?
Talking real time, though, basically, whether the technologies will be there.
And I guess, I could see that, and it's really just issues of privacy. What level of privacy are we willing to compromise? Personal privacy.
In the back?
Yeah, this is a question to David Relman. So we're talking about stability of the human microbiome and transitions between different stable states and within the context of diseases mainly. So my question is, do you think it might be a possible correlation with the transitions between these states and aging in such a way that with aging you could have more easy transitions between different states?
Yeah, there's a little bit of work on aging over these relevant long periods of time, but not a lot of data, and yet theoretically it to me it makes a lot of sense. I mean, even to think about changes in those landscapes that correlate with the process of getting older and the accumulation of previous disturbance events, which we know can have a compounded effect, accumulated effect over time. So yeah, I think it makes a lot of sense to think about that as a setting in which to design experiments to test whether the predictions would hold.
So I have a question related to a previous question, one for Julia, one for Larry. So for Julia, so I think it's a part of a rhetorical question, like how could you replace patient when it actually was released? Because you couldn't discover, you couldn't detect the microbe, right, the infectious microbe in the body. So are there procedures you envision would allow sensitive detection of these ones so that the patient will not be released until it's actually absolutely gone, so that it will not start the outbreak of infection?
David can answer that more. I mean, it's really, they already do things like that when you're I mean, not when you're talking about KPC, but when you're talking about, should you start administering antibiotics? And, clinical micro labs are all about how fast can you get this test done with what sensitivity. So those things are driving, those exact questions. Can I get this done before the patient is released from the hospital? That is absolutely driving all of clinical micro already, and at what cost? So, yes, because it's actually, what we're talking about, you swab inside someone's nose and within 16 minutes, you know whether or not they're an MRSA carrier. Same way you could, do a rectal swab and know if someone was KPC positive in the same time, turnaround time. I mean, clinical diagnostics is totally going in, is totally already in that direction.
I guess a related question for Larry is that I really enjoyed hearing how you checked your personal health. So do you envision, like in the very near future, I mean, everybody could do that. So for example, you have a test, some test paper, for instance, right, for blood, for stool, and the data will automatically be downloaded into computer and getting analyzed. So how soon do you think this would happen?
I'm counting on Lee Hood to come through with his vision of, I just gave 12 vials of blood last week and with 6 different needle sticks because the first person wasn't very good. And so when Lee tells me he can do 2,500 proteins with a single drop of blood, I'm all for it. And that, of course, drives the cost way down as well. So I think the kind of things Lee has been talking about for a decade of using the emerging nanotechnology, the amazing microfluidics you saw on the tours upstairs, so that you can begin to do that. The separate thing is what's going on in the cloud. So, every night I wear a Zeo headband with a tracker that every 30 seconds measures whether I'm in light, deep sleep or REM or awake, and then graphs it next to my bed so I'm watching during the night. I get into sort of competitive sleeping. And then that's all uploaded to the cloud. And so there's a startup that has, tens of thousands of people with every 30 seconds all night long. I've been doing it for a year. And that's hundreds of times as much data as all the sleep studies ever done in academia put together. And it's in some startup's cloud. So one of the things that we're going to have to get at is how do you get interoperability of the data? It's the same question as you have in the social network world. Facebook wants to keep you inside Facebook. Those are your friends. You should be able to move that structure of your friends to any other service, but that requires an open API to do it. Same thing should happen for, it's your data. If your body generated it, you own it. That's the way I view it. And then you should be able to move it from one place to another, and you're going to have to do it to do the mashups you need to do between one kind of body data and another kind of body data. But so I think a lot. I would say certainly 10 years. This is all going to be true. In 5 years, you're going to be more than just the early adopters.
Sorry.
Yeah, that sounds pretty cool. I've heard that somewhere.
Great. This is a question for Julia. I'm wondering about the strain typing assay you described that uses whole genome sequence technology. And I'm wondering, can you talk about the utility and the potential future use of this in place of PFGE for strain typing, or perhaps in at the same time as using that? I know public health is PFGE is the main strain typing technology now, but what do you think about systems biology infiltrating that?
Yeah, I mean, it was actually kind of remarkable to me when I came into a clinical micro lab and found that they still used pulse field gel electrophoresis because, I remember building one in graduate school, and I really didn't know that people still use them. And it made me feel great to have some piece of lab equipment that I actually knew how to run. But, on the other hand, I also am not used to that probably for a reason that, moving over to PCR-based assays is just, and digital information is so valuable. So I really think that any field in medical practice that doesn't use digital information, that needs to communicate among different departments and between hospitals, I think using some form of digital information is incredibly valuable. When I think about what parts of medicine are really open for genomics, a lot of people have talked about pathology and so on. I actually think medical microbiology is just, they are ready for genomics, and if you change the chief of medical microbiology, he or she will then change every other department in the hospital because they're one of those key leverage points in a hospital. So I think a lot of people are excited about moving over to genomic data, and systems biology data is really great for them to have some sort of, the medical microbiology already has that, where they're looking at antibiotic resistance profiles, they're looking at, they look at different kinds of information to classify, and genomic information is a type of information that microbiologists are, ready to handle. Optical maps is the other thing they use. Yeah.
Okay. Let's maybe take the 2 remaining questions.
Thanks. This question is for any of you, really. A lot has been said about the promise of this metagenomic data as a diagnostic biomarker, but really, as it has been alluded to, the etiology of a lot of these autoimmune diseases is still really not nailed down. As I read a paper recently where there's a dramatic association was shown between ulcerative colitis and skin infections such as paronychias, and as far as I know, there's no explanation really for some of these associations. So I'm wondering if, now that we have all this microbiome data from multiple body sites, skin, gut, oral cavity, if you see any role for that data in elucidating the etiology of some of these illnesses, or if ultimately that's just going to be a question that immunologists say have to answer.
I think there definitely is. If you take what Mike Snyder has done and you add back in the genome of the microbes, and you were doing that monthly movement, he saw his type 2 diabetes turn on, which he didn't have before. He had a genetic predisposition to it. And he could see it actually, all of these things, in these tens of thousands of things he was tracking, turning on from a systems biology point of view. And I think the only way the pathogenesis of IBD or other autoimmune diseases are going to be understood is precisely doing this sort of thing, where you're taking the time series of both the human and the microbe, and then you just watch it happen. And we haven't ever done that. That's why they keep saying you don't know which is the chicken and which is the egg. And by the way, the skin disease, if you have like psoriasis, I have the same markers for like HLA for psoriasis as I do for this, and it's quite common for people with IBD to develop a lot of other autoimmune rheumatoid arthritis is quite common because basically if it's the gut microbes dysbiosis interacting with the human, as Craig Venner said the other day, 1/3 of the metabolites in your bloodstream are produced by the gut microbes. So, it's going to go all over your body.
The key is going to be to have the kinds of time series data that precede the development of these clinical events, because there are almost certainly going to be alterations at the time of pathology, and you'll never know, which came first in causation. But the other thing is we need a lot of each of these different kinds of time series with the same standardized metadata, because the statistical power in an N of 1 or 3 or 6 is insufficient. So that's just where we need to be.
Okay. Last question?
Thank you for your thoughts. In terms of antibiotics and in the spirit of personalized medicine, and since we are 10 times more microbe than human, do you think that we're doing ourselves a disservice that when we do have an infection that we're treating ourselves with a broad spectrum antibiotics that likely are having detrimental effects on majority of the commensal organisms?
The problem is that diagnostics has not led and it has followed, and so without the ability to recognize a specific microbial agent as the possible cause and exclude the possibility of lots of others that could also be there, you're stuck with empirics until you know what it is you're treating. And so the drug development enterprise has been, stuck in a blind state with, the need to develop a broad spectrum agent. It's also a much more revenue generating strategy as well. We're in a kind of a dysfunctional little reinforcing loop that we have to break out of.
And I would say if you take into account that 80% of antibiotics in this country go into animal feedlots and then into the ground supply, into the water, and into our bodies, as Eric was showing, by eating the meat, this is insanity. I mean, and you add that to the promiscuous prescription by doctors who don't want to be yelled at by their patients. They need to get some backbone and only provide antibiotics when it actually is needed. And now, when you have a life-threatening thing and you need the antibiotics, by all means have them. But when you look at how long-term the impact is on your microbes, and they're just beginning to really, there are a number of papers that are pretty good, but it's just really beginning to be studied. This is your whole life you're talking about, that you're having these impacts over with repeated use of these. In fact, one of the big problems is they're trying to find people for fecal transplants, which I think is a very interesting possibility because you bring over the entire biochemically linked community in inserting it, so to speak. And they want to find people that can provide stool that haven't ever had antibiotics and they can't find any. Because in fact, is Jonathan Eisen still here? He was saying that even in the Amazon jungle, some of these native people, the water in the Amazon from the runoff from all the ranches that are making cattle is so full of antibiotics that they are exposed to. I mean, somebody needs to sit back and think a little bit.
All right, let's break for just about 10 minutes, and then we'll come back for our keynote closing remarks by Pierre Bourque. Thanks to the speakers again.